My story is about mutants, not zombies like in The Walking Dead, but powerful mutations that are genetic changes in the DNA that you and I should care about. And that's because all of us right now, we're all accumulating changes in our DNA, changes that help the cells in our body to adapt and thrive upon changes in the environment, which can be very good for survival. But sometimes it can also be very bad news. For example, bacterial cells, viruses, all these disease-causing pathogens can also accumulate very rapidly these changes in their DNA. And that's been shown, these changes have been shown to help them to thrive and survive when exposed to very stressful conditions, like, for example, when they're exposed to a drug. This idea of evolution of resistance has been really threatening our modern medicine. And so my research focuses in one fundamental question. Can we detect all of these possible mutations that help a pathogenic cell to resist to a drug? But because drug resistance is really impossible to predict, we took the approach of instead engineering all possible mutations in a target gene of interest, and then design a competition experiment so we can find out which are these resistant mutations. It's like a race, like the Vancouver Sun Run, for example, where a group of individuals are to run together in parallel. Some have great stamina, others are better at sprinting, but really the winners of the finishing line will really depend on the running route that they are registered for. And that's exactly what's done in my experiment, a competition experiment where a mutant population made of over 20,000 cells with different mutations were challenged together to survive under lethal drug conditions. And then for practical purposes, we use the genetic model Baker's yeast, the yeast that makes your beer. And using the next generation sequencing technology, we can then identify very quickly what are these leading mutations that can help these target genes to adapt and outcompete the rest of the mutant population. Summarized in this cartoon on the right of the slide is the summary of my results, where a case study where I use a potent drug shown in yellow that can bind very tightly to its target protein shown in orange. When we expose the mutant population to this drug, we found lots of different mutations mapped here in blue. Some were unknown, some were published in and observed in patients, but all to say that drug resistance is really complicated. And our method can identify all these genetic mutations that can render a drug ineffective. And what's more, our method is widely applicable to all kinds of drugs. So we're hoping with this strategy that we can find one, better drugs, and two, better diagnostics for drug resistance. Thank you very much for your attention.